I'll start again. Good morning, everyone. Welcome to today's seminar. To the people online, please put a thumbs up if you can hear us well and put your questions on the QA. Today we have Eric Garnier. I will let Cyril Viol introduce him for us. Okay.
So good morning. So I'm really happy and proud to introduce Eric Garnier today from the CEF at Montpellier. So I will say a few words in French because this is a special moment for our community, for Eric, of course, for me also, to be honest. As like for others in the past, for instance, Finn Kleshberg, Eric will take us through the main stages of his huge career, alongside with the development of functional ecology. So functional ecology is a broad, vast field, often broader than we can imagine. And I hope that at the end of this seminar, we know a bit more about what functionality is, depending on what Eric thinks about it. Donc, comme je vous le disais, je vais dire quelques mots en français aujourd'hui, puisque c'est un moment assez particulier aujourd'hui pour nous, à quelques semaines, semaines de la retraite d'Eric. Eric va nous retracer une partie de sa carrière qui s'est faite de façon concomitante avec l'explosion de la discipline de l'écologie fonctionnelle, explosion qui s'est faite par palier ces 30-35 dernières années. Et c'est concomitant, mais c'est pas fortuit non plus, puisque Eric a énormément contribué à l'écologie fonctionnelle à l'échelle française et internationale, en tandem avec deux personnes, Catherine Roubaix qui est dans cette salle et Marie-Laure Navas qui doit être en ligne. Donc voilà, Eric Garnier est directeur de recherche au CNRS. Il a commencé, donc il est diplômé de l'agro de Montpellier, donc il est ingénieur agro. Il a commencé par une thèse en écophysiologie, j'ai envie de dire pure. Il a soutenu en 1985 sur des indicateurs de stress hydrique chez le pêcher pour savoir le niveau d'irrigation du pêcher. Donc, c'était la physiologie et des sciences agronomiques. Il s'en est écarté assez rapidement jusqu'à y revenir plus récemment avec des gros projets sur l'olivier, notamment dans le bassin méditerranéen. Donc, la boucle est bouclée, la sécheresse méditerranéenne est pas loin. Eric est une figure de l'écologie en France, clairement, et j'ai envie de dire une figure de l'écologie à la française, si j'oserais dire. Donc, il s'est énormément impliqué pour l'écologie et pour la communauté des écologues, avec énormément d'investissements collectifs. Déjà, à l'échelle locale, il a été pendant longtemps directeur des études du DEA B2E, qui est connu aujourd'hui sous le nom de Parcours Darwin. Ça a été aussi un temps le directeur du LabEx CEMEB, que beaucoup d'entre vous connaissent. Il a été aussi le président, et c'est pas anodin, du comité d'organisation du colloque Ecologie 2010, qui a été le premier vrai grand colloque à l'échelle nationale d'écologie, qui a contribué beaucoup à fédérer toute la communauté des écologues à l'échelle française, et maintenant aussi des évolutionnistes au sein de la SFE au carré. Et ce moment a été réellement important pour notre communauté d'écologues en France. Il a aidé beaucoup de choses. Il a créé, était directeur de 3 GDR, donc un GDR sur les traits fonctionnels, un sur l'éco-informatique, j'y reviendrai un peu après, et un GDR aussi international avec le Québec, le Canada, avec son compagnon de route Bill Shipley. Et aussi et surtout, Eric a initié et créé, en collaboration avec Xavier Leroux, un outil que beaucoup d'entre vous connaissent aujourd'hui, c'est le CESAB. C'est le Centre d'analyse et de synthèse de la biodiversité. C'était totalement novateur pour la France. C'était connu notamment aux États-Unis, mais totalement novateur pour la France. Et Eric l'a créé depuis le début. Je me souviens même de discussions à la cantine où il discutait du choix du carrelage dans les toilettes. Donc, il l'a créé du début à la fin. À l'époque, le CESAB était à Aix-en-Provence. Et effectivement, ça a donné une impulsion énorme aux recherches en écologie en France. Donc voilà, parmi beaucoup d'autres choses. Je ne sais pas si vous avez vu les chaînes de médias en continu, mais il paraît que la France est envahie en ce moment. Est-ce que vous savez par quoi? La réponse est plus consensuelle que la question. Des fake news déjà, mais sinon, il paraît que la société Chine envahit effectivement la France, c'est vrai. Et il y a eu beaucoup de débats entre l'opposition entre Chine, donc la fast fashion, et la haute couture à la française. Je trouve que ce débat caractérise assez bien Eric, parce que pour moi, Eric est réellement l'anti-Chine de la science. C'est clairement plus la haute couture à la française, à plein de niveaux, à plein d'égards. Déjà, en tant que couturière, couturier, pour ceux qui connaissent Eric, soit dans les mesures, soit dans l'écriture des articles, bah, c'est une rigueur et une minutie qu'on ne croise nulle part. Au millimètre carré, lui qui refusait parfois justement la technologie, parce que voilà, un planimètre à l'ancienne, où on manipule l'intensité de façon fine, bah, on va avoir une précision réelle de la mesure. Ça, c'est Eric. Dans l'écriture des papiers, beaucoup d'entre nous ont été formés plus que jamais à l'écriture. Chaque phrase sont pesées, travaillées. Et donc ça, c'est la rigueur et la minutie de la haute couture menée par Eric. Quand on parle de haute couture aussi, on pense évidemment aussi pas qu'aux couturiers, mais aussi aux créateurs. Et clairement, pour moi, Eric est un créateur de mode, un créateur dans sa discipline. Donc, plus que suivre la mode et suivre le vent, il a clairement créé la mode, été le vent, pas nommé de vent parce que je ne suis pas aussi bon navigateur que lui. Donc, c'est pour ça que il l'a créé à différents niveaux et je crois que ça va être les trois grandes parties de sa présentation, donc je vais les citer rapidement. La première, c'est justement le passage de l'écophysiologie pure, entre guillemets, à l'écologie comparative pour comprendre la biodiversité dans son ensemble, sans perdre son âme, sans perdre les détails, sans perdre les mécanismes. Et ça a été le défi à l'époque, et c'est toujours le défi, de ne pas faire cela. Et Eric, clairement, a été pionnier sur cela. Et il va évoquer ça, donc de passer aussi d'une espèce où on comprend très finement une espèce à plein d'espèces à la fois. Une autre un autre point où il a été pionnier, c'est une fois qu'on comprend ces organismes, ok, on décrit bien la biodiversité, mais comment on passe au fonctionnement des communautés, des écosystèmes, donc ce qu'on appelle un changement d'échelle, en anglais un scaling up. Il y a toute une approche méthodologique, conceptuelle et théorique, où Eric Garnier, aussi en collaboration très étroite avec Sandra Laborel, ont été clairement pionniers, notamment par le développement pas que, mais de ce qu'on appelle le cadre conceptuel du réponse-effet qui a envahi l'écologie fonctionnelle depuis. Et enfin, Eric a été avant-gardiste, et franchement, je n'aurais pas mis un billet dessus, honnêtement, connaissant Eric, une critique, mais au contraire, c'est sur l'aspect éco-informatique, la gestion des données. Il était pas mal réfractaire à pas mal d'outils et de technologies. Et bah, franchement, c'est un respect absolu parce que quand on le regarde, on dit on va y aller, quoi. Parce qu'il a été réellement avant-gardiste sur le travail sur les données, l'organisation des données en écologie en général, pas que en écologie fonctionnelle. Travailler avec des informaticiens, comprendre leur vocabulaire, leurs concepts. Il a même encadré en encadrant principal une thèse en éco-informatique. Il a développé une ontologie des données en écologie fonctionnelle. Ouais, c'était un peu et c'était clairement avant-gardiste que personne, nous dans l'équipe, quand il nous disait oui, il faut construire une base de données relationnelle, tout ça, on le regardait avec des yeux, on disait ok, peut-être. Et même au niveau CNRS, puisque dès qu'il a demandé même un petit peu de financement, il n'avait rien eu sur ça. Alors aujourd'hui, le CNRS et tous nos organismes ne jurent que par ça. Ils ont créé des unités entières sur ce sujet. On ne peut pas soumettre un projet ANR sans avoir un plan de gestion des données. Donc, à tous égards, il a été avant-gardiste. Et c'est vrai que quand on parle de l'anti-Chine, c'est à tous les niveaux. C'est sur ça aussi, effectivement. C'est sur le fait que je pense que peut-être à quelques semaines, même s'il restera un peu, j'espère, avec nous, de sa retraite, je pense qu'il y a une petite frustration, voire de désarroi, de voir effectivement cette science un peu fast science exploser, notamment dans notre discipline. Et je voudrais juste finir sur Eric au CEF, donc c'est un peu le sage du CEF. Mais le sage, depuis, pas une question d'âge, parce que ça a toujours été, son pas de porte est assez mythique, parce qu'il y a toujours quelqu'un à son pas de porte pour lui demander des conseils, que ce soit des tout jeunes étudiants qui commencent ou des chercheurs confirmés. Et ça fait toujours du bien. On part toujours avec des livres des années 50 ou 60 en disant « Ok, ce qu'on avait imaginé, c'est déjà fait, et mieux, mais c'est pas grave. On va essayer de les lire, mais ne les a pas toujours lus, mais au moins on a un peu Eric en garant. Donc merci de nous rappeler que la science, c'est toujours ce qu'il nous dit au quotidien, a commencé avant les années 2000. Donc Eric, merci beaucoup pour ta sagesse, effectivement, ta rigueur, ta créativité. Fait que tu sois un phare pour nous. Ouais. Merci beaucoup pour tout ça. Et puis, on organise une journée scientifique autour de toutes ces questions le 15 janvier dans l'amphithéâtre du CNRS. Donc, vous êtes tous bienvenus. Donc, voilà, et qu'on me dise the floor is yours.
Thank you, Cyril, for this impressive introduction. Thank you. I don't know whether I'll be at your expectations. We'll see. So anyway, as Cyril said, I'm going to take a few flashes of what I've done during this 35 years, more or less. Once I'll begin with the context with which I started the research in the 80s, 1980s. Then I'm going to switch to the focus on the organism level. What is it that we talk about when we talk about threats? Why we do that? And then, as Cyril said, we are going to say a few words on the scaling community and ecosystem with a response and effect framework, and we'll feel that afterwards. Making sense of the data, which is quite interesting. I think a few closing remarks. And I've made a choice, a very strong choice in this presentation, is that I've decided, rather than showing many things, focusing on a few things and try to figure out and explain to you the mechanism, the approach by itself, rather than putting a lot of results all together. So in terms of context, so I was hired in 1988 by the CNRS, and it was the very year when this book appeared, which was edited by Edward Wilson, which is called Biodiversity. And this is the first time the word biodiversity appeared in the literature. And it was this book was edited after a conference, which was held in 1986 in Washington, to warn people, to warn the political world that biodiversity was in danger. So they created the word biodiversity at that time. So it was created in 1986, and it's the first time there was a book published on biodiversity. And at that time, the biodiversity was defined as the diversity of life forms. Very broad definition. And I was hired in 1988, and the head of the lab at that time was Francesco Di Castrit, but some of you probably know. Francesco made a very profound changes in that set. Set was called before Centre d'études phytosociologiques and Ecologie. It was really focused on phytosociology for a long time. And Francesco was hired actually to change the whole thing. And he said that, decided that three things were very important for the set. It was biodiversity, the first one. The second one was global change. And the third one was sustainable development. And I think we still are more or less along these lines, 38 years after. And one of he wrote a lot of things. Francesco was an entomologist in the first place from the Mediterranean basin. And in a paper he published in 1990, he said that biodiversity is becoming the most exciting and challenging topic of modern science. So it means that it's becoming. So it means that really we are in the process of getting to what is biodiversity. We didn't have we knew what biodiversity was in terms of number of species. We knew that before, but I mean it's really becoming it's becoming a scientific object to study. And this is 2 years before the Rio summit in 1992 with the Convention on Biological Diversity. And right from the start, the idea was so this is a more modern representation of what actually was said at that time. The idea is that they were a component of global change that affect biodiversity and ecosystem functions directly or through biodiversity. And this whole thing would affect ecosystem services that the ecosystem deliver to human societies. So that was already the background. This is a more modern way of seeing it, but it was already the case when the Convention on Biological Diversity was launched. So this is the background from which I started with And so there was a lot of work during the 90s. And there was a very influential issue of Science, which was published in 1997, with a lot of things on global change, the impacts of global change. What is global change? There is a very important paper by Peter Tusek, who actually explains what global change is. And I take this paper, I take this framework from a paper by Terry Chapin in this issue. And this is more or less the framework that I've been working with in the last 30 years. So the idea is that there are components of global changes, which are land use, biotic production, atmospheric composition, biochemistry stuff, which affects the biological component of the Earth. And the idea of Terry Chapin is that this is affected through biotic functional traits. So the idea is that these global changes affect biodiversity through functional traits, and these functional traits are linked to ecosystem processes and services and community processes. So as you see in this slide, the effects on ecosystem processes, the processes doesn't go directly from species composition, but from functional traits. So that was the very first, I think, paper which expressed that directly. So for today, I'm going to concentrate on what are biotic functional traits, which ones are important, and on the effect of functional traits on ecosystem processes. I'm not going to talk about response in the end, because that would be too much important. Okay, so the idea is the core thing is that we have to characterize the organisms we are working with functionally. That's the idea. And I'm going to focus on plants. I'm a plant experimental ecologist. I didn't do much on animals or other organisms. So I'm going to concentrate on plants. But actually, many of the things that we do on plants actually now are currently being doing on animals and on microbes as well. So the idea is to switch from a taxonomy or phylogenetic view of life on Earth to an approach with traits, the traits of plants, which actually gives us how the plants, the organism interact with their environment. If I give you the position of a plant, a particular plant on the tree of life, you actually don't know how it's going to interact with the environment. The idea is that if you use the traits, you actually have a common currency for functions. And for example, this tree is going to produce shade, which is going to interact with its neighbor very differently from these herbs, these grasses, herbs, which are going to interact very differently on the basis of simply because they are bigger. So they are going to make more shade. And if you measure the height of the plant, you're going to have more information on the interaction between the plants and the environment rather than saying where it is on the phylogenetic tree. So that's the basic idea of the traits. We describe the interaction with the environment, the abiotic environment, and the biotic environment. So once this is said, what do you do? I mean, you could measure potentially thousands of traits of any organisms. So the idea is to construct matrices like this one of species trait species trait matrices, and which trait do you decide to put in? Which one are relevant? Which one are relevant for the plants? Which one are relevant for the ecosystem processes? So that's the key question that we had. And in 1990s, we didn't know exactly what to do. We had some ideas already, but not very clear. So the idea is that we developed some kind of comparative ecology, which was already done by Phil Graham and Peter Reich, for example. So the idea is to have these traits measured for a wide range of species. This is very different from what we call the haute ecology, which is knowing everything on a particular species, and then you could measure everything on a particular species. But this is not tractable for a high number of species. So the idea, which traits do you do to do that? So we have a number of criteria to choose these traits. The first one, it has to be linked to a plant function. If there is no function behind, well, don't do it. You need to have substantial variation for the values of these traits. If you don't have variation, if all the organisms have the same trait values, then you're stuck. A very important point, functional ecology and comparative ecology has been really criticized for that, is that we have to choose traits which are relatively easy and quick to measure, because we have to measure them on a large number of species. We are studying biodiversity, so we need to have that on a large number of organisms. When I prepared the concours from chargé de recherche to directeur de recherche, the head of the section was Patrice, David, the guy who was working on Drosophila, Jordan. And I finished my talk, my presentation, and he said, "Wow, how can you work with more than one organism?" Well, then, yeah, didn't have the control.
How can you work with
Sorry. So it was a bit tricky. So and a lot of people criticize that because we degrade the information. That's we are obliged to degrade the information and have less precision than if you do something on autoecology. And then it has to be measurable in standardized protocol. We'll come back to that later. So it was the criteria we had in mind when you start to do these things. And then the idea is that traits, not all traits are relevant. And we use this framework with Cyril. So Cyril has been writing during his PhD thesis a very important paper, which is now very highly cited currently. And and the idea is that we need to have traits that are related to the performance and the fitness of the plant. So that's a scheme which seems pretty straightforward. And the idea is that we have these traits, which we call functional, either morphological trait, physiological trait, or phenomenological trait for FDP. And these traits are correlated with one another, or not. And the idea is that they should be linked. If you have to select them, they should be linked to at least some index of performance, such as vegetative biomass, reproductive output, plant survival, which is called plant performance, and this is linked to individual fitness. So the first idea of this selection is that it has to represent a relevant function, and it has to be linked somewhere or another to plant performance. Okay, which traits? That's one of the things we think is that vegetative biomass actually represent quite, at least to some extent, the reproductive output and maybe plant survival. So I'm going to focus on how you produce vegetative biomass. And you can produce vegetative biomass at the end of a growing season, which is here. You can do that three different ways. You have differences at the end of the season because you have differences at the beginning of the season. So think of an annual plant. As an annual plant can have different seed sizes. And if you have bigger seed size at the end, if the growth rate of the plant is the same, you end up with having more biomass in the end. So that's one possibility. The second possibility is that you start with the same starting point, and you end up with different biomasses at the end because you grow at different rates. So that's the second option. And the third option is that you end up with different biomasses because you have the same initial size, the same growth rate, but you start growing at different times during the year. That's basically phenology. So this is basically seed size for initial size, growth rates, and phenology. So you can write that very quickly in a very simple equation. So the biomass at time t1 is equal to the biomass at time t0 multiplied by exponential relative growth rate, which is the growth rate of the plant divided by its biomass, and it's an indicator of the physiological functioning of the plant. And the last bit, which is T0.1 material, basically the phenology of the plant. How long does it grow? So if you do that, you actually tend to see kind of traits that we are looking at. We are looking at traits which relate to plant mass or size at a given time. You are looking for traits that are important for initial size. You are looking for traits which are important for physiological processes and traits which are important for phenology. So that's basically more or less the idea that we followed. Maybe it's a backward construction, but in the end, that's how it is. So now, okay, this is set. What do we have in stock in our storage stuff to see how these traits vary, whether they are vary altogether, if they co-vary, yes or no, and how do they vary across the the plants in the world. So we used, there was an initiative launched in the mid 2000 years, which is called TRI, which is a global network of scientists, and it's also a plant trek database, which involves a number of participants. And TRI is not an acronym. When it started, it was really the idea of we are going to try to get data together. That was really the objective of that. We didn't know whether it was possible. So it was held by run by Jens Katke from the Jena Institute and Sandra Diaz in particular from Argentina. So we ended up with actually it worked. So we tried and it worked. So there is quite a number of data. Currently, this is the release 7 from a TRI. And this is the type of traits that you have in the database. I'm not going to go through that in much detail. But the idea is that, to answer our first question, is that we used 6 traits, which were for which we had the complex matrix in the TRI database, which is a little more than 2,000 species. And the 6 traits are adult plant height. This might be one indicator of size that I told you before. Diaspore mass, which would be the starting point for the growth. Stem specific density, which is an index more or less of the woodiness of the plant. It's really related to wood density, in particular in trees. Leaf area, which is an index of the size of the leaf, and 2 other. So this is more or less morphology, I would say. And we have 2 other traits. I'm going to go back later on leaf mass area, which is maybe I can use. Okay. Thank you, François, for reminding us. So leaf mass area is the area of a leaf per unit leaf mass. Sorry, it's the reverse. It's the mass of the leaf per unit leaf area. So here you have a sheet for which you have a certain leaf mass per area. And if you hold this sheet, now it's the same weight, but it's twice smaller. So the leaf mass per area would be higher. So it means that the structure of your leaf is going to change very much when you change the leaf mass per area. And nitrogen per leaf nitrogen per mass is the other one I'm going to do. So what we see is that there is a strong structuration in the phenotypic space with these 6 traits. This is the results of the principal component analysis, which was run for all the species. And you see the first axis here is linked to leaf area, seed mass, diaspore mass, and adult height. Basically, this first axis is a size axis. And what you see, this is the density, the color of the spots indicates density of the points. So you have a high density of points here, which are basically trees, and a low, large height. And most herbaceous species are around here. So this is more or less a distinction between herbaceous and woody species. But this goes with differences in seed mass. So there is a tight correlation between height and seed mass. Specific stem density, to be high, you need to have a dense wood, either a dense wood or a large diameter. And leaf area, which is another size. So that's the first axis. And this first axis, the size axis, is actually linked to many things in terms of the plant. Here you have the results from a series thesis, showing the relationship between when you have monocultures with different height. This is the light interception that you have at the soil, at the level of the soil. And when you increase the height of the canopy, you decrease the light available below. It means that if you have a competitor going in, it's going to be the competition will be much higher in this range of height as compared to this one. So it's very strong effect on competition. The second one is that it is another estimate of leaf size, of plant size, which is leaf area, the total leaf area of the plant, which is tightly linked to the fecundity. This is the reproductive output, which is tightly linked to the fecundity of the plant. So again, the size axis is linked to reproduction, one way or another. And then it's also linked to seed dispersal. This is the relationship between the mean distance where a seed can go, as a relationship from, so there are different terms. Height is here, height, This is also a function of wind speed. If the wind is very strong when the seed disperses going further away, and this is the term which other traits which represent the speed at which the seed goes down when you throw it, not throw it, when you drop it. Thank you. When you drop it. So size really has many impacts on many aspects of plant ecology. The second axis here is structured by leaf nitrogen per unit mass and leaf mass per area. So the two terms which are just presented here. So it represents what we call the leaf economic spectrum, from leaves which are called slow and tight, that is, they photosynthesize slowly, but they conserve the resource efficiency, and fast and leaky, which are the reverse. And to go a little further on that axis, we actually had the first compilation of data, which was called during the BlockNet network and database, which were actually focused on the carbon synthesization, which measured on more than 2,000 species, the photosynthetic rate, leaf mass per area, and nitrogen per mass. And one of the main results of this study is that there is a very strong relationship between photosynthetic rate, leaf mass per area, and nitrogen per unit mass. The relationship is negative with leaf mass per area and positive with leaf nitrogen per mass. And here you have photosynthetic rate. Remember the equation I showed with the relative growth rate. To some extent, it's not perfect, but to some extent, leaf photosynthetic rate is related to relative growth rate. So that's the physiological component that we were looking for. So why are these related? So the leaf mass per area, as I told you, is the ratio between leaf mass per area, and it tells you something about light interception by the leaf. And leaf nitrogen content per leaf mass is actually tightly linked to the photosynthetic processes in the leaf. This is the allocation of nitrogen in the leaf. And you see that 75% of the nitrogen in the leaf is connected in one way or another to the photosynthetic process of the leaf. So these two traits, actually, you just remember your first year university courses. This corresponds to actually the two reactions of photosynthesis: the light reactions for leaf mass per area and the dark reactions for the leaf nitrogen. So it really describes the process of photosynthesis quite nicely. The other very important results that we had with this calculation is that there is a negative relationship between the net photosynthetic rate I just showed you and the lifespan of the leaves. Time the leaf remains on the plant. And this tells you that some of these species, which have a short lifespan and a high rate of photosynthesis, have a fast rate of carbon acquisition with high leaf mass per area and high leaf nitrogen content, which are opposed to leaves with an efficient resource conservation with a high leaf mass per area and low nitrogen content per unit mass. So this is a physiological trade-off, which is very widely recognized now. And it tells you that there is an envelope, which is actually, it shouldn't be seen as a very strong relationship between the two traits, but as an envelope. Here, you would not be possible to construct a leaf with these characteristics, with a long lifespan, with a fast rate of photosynthesis for many reasons. And here, you would have a low photosynthetic rate with a high leaf lifespan, and it's not efficient either. You're going to be completely out if you do that. So it should be seen as an envelope of what is possible and what is true. This is interspecific. And because we are in Montpellier and we are a very strong place where evolution is. Cyril, when I prepared this talk, Cyril told me, hey, you should say something about intraspecific variability. Because I know that as Drosophila, maybe we should work on only one dimension. I don't know. Anyway, you can zoom in. We can zoom in certain cases. We can zoom in this space. And the first one, if I use the global spectrum of plant form and the first PCA, we have data in the group when Sandra Diaz came and spent a couple of months with us, and she actually produces this slide. So this is the global spectrum, the one you show. And this is what happens when you plot stuff for Arabidopsis thaliana. This is data from François and Cyril. This data which you plot when you plot durum wheat, durum wheat. And this is what you do when you plot olive trees, the olive tree, Olea robea. And what you see here is that Arabidopsis is just amazing. There's an intraspecific variation across gene types, which is very impressive. But as you can see, it changes only on one dimension. It changes on the leaf economic spectrum dimension. It cannot do anything about height, which is Arabidopsis is stuck with height, cannot grow bigger. It's just it's one of the axes which changes. And you see it's more or less the same for wheat, little less the same for tree, for olive tree. And this can be the case because as you have wood, you can probably modulate more what you do in terms of height. So this is what we have for the global spectrum. If you do the same for the leaf economic spectrum, for Arabidopsis and Olea robea, then the relationship between leaf mass per area and net photosynthetic rate is significant in both cases. So even if you zoom in, you can see the same process at work. And this is probably because leaf mass per area is a key trait to capture light. But if you look at the relationship between net photosynthetic rate and leaf lifespan, in Arabidopsis, you still have the relationship that you showed at intraspecific level, but it's not the case anymore for olive. So I think this relationship, this covariation, are tighter when you have a strong mechanism which is behind, but it's not the case if you do that for when you don't know exactly the mechanism. So if I conclude on that, there are two major functional dimensions which are identified, the plant stature and resource use, but we are pretty aware that the phenotype is actually multi-dimensional, much more than these dimensions, and you can think of many dimensions on the function of the different organs, which you can see here. But the question here, how many axes do we need to actually describe properly and functionally a phenotype? And this is the main question. We don't know. And the other results in the selection pressures might vary at the macro and micro evolutionary scales. And the idea is that relationship which has concerns at all levels reflect strong mechanistic basis, which is probably not the case when the relationship breaks down at the final taxonomic scales. Okay, so I'm going to switch to the second part. So the idea now is that now that we have ideas of how to describe an organism functionally, with Sandra Lavoraille, we developed a conceptual framework in which traits are involved in what I showed you in one of the first slides of the talk. Is that the idea is that the changes in the environment, so that might be global changes, but any kind of environment, affect ecosystem functioning and community structure through response traits, through traits. Sorry. The traits that can explain the response to changes are called response traits, and the traits which are affecting ecosystem functioning and community structure are called effect traits. So if I explain a little bit what we show here, what are community structure and diversity. In a plurispecific system, this is the distribution of trait values in a community. This is the overall distribution of traits of the community, which results from the distribution of traits within species in this community. So this accounts for intraspecific variability, but what you are looking at is the results of these changes, of these differences. And you can have different metrics to characterize this structure. The first one is the community average, the average value of the traits of the community, and trait variation, which is called functional diversity, which may be any metrics. I'm just going to use two metrics, but you can use quite a number of metrics to describe them. There's a little bit of confusion in the literature, saying that functional diversity is the whole thing. And I think it's not very well done. I think we should really distinguish these two, which are different aspects of the community structure. So, and these affect ecosystem problems. So now there are two main hypotheses for the scaling of from organisms to ecosystem. The first one is the dominance of selection hypothesis. And the statement is that the traits of dominant species influence ecosystem properties. And this is called the biomass ratio effect hypothesis, which was formulated by Eric Garnier in 1998. And the prediction is that ecosystem properties under this hypothesis depend on community-weighted mean of traits, that is the average value I showed on the trait distribution. And the other hypothesis, the complementarity hypothesis, is the idea that more dissimilar species will allow a more complete use of resources, and that the more complete use of resources will enhance the rates of ecosystem processing. And then the prediction is that higher functional divergence or functional diversity will be positively related to rates of ecosystem processes. So these are the two main hypotheses for which traits are related to ecosystem processes. And these are not mutually exclusive. You can have one and the other. So we tested that. So I just take one example. We did that for a number of traits and a number of processes, but I'm just going to take one example from the work we've been doing in Lafarge, which is a rangeland on the Larzac plateau, the plateau Larzac, which is grazed by sheep. It's very close to Montpellier, 30 kilometers from Montpellier. So it is a domain held by INRAE, and it's a 300 hectare domain which is grazed by sheep for meat. It's not for crops. No? And there are 3 treatments were applied since 1978 there. One treatment is the classic stuff which is on the Larzac, which is grazed and fertilized with a low grazing rate. One which is ungrazed, unfertilized, so they put fences so that there is no grazing there. And then the third treatment, which is intensely grazed and fertilized. There are 3 main treatments there. And I'm going to test these different hypotheses for relationship between traits and ecosystem processes for dry matter digestibility. Digestibility is a synthetic measure of the amount of energy which is available to herbivores in plant constituents. There are 3 main important things for herbivores: the amount of biomass which is production, the digestibility of this biomass, and the timing at which it is available for grazing. So I'm just going to concentrate on digestibility. So we measured species abundance in these three treatments. We measured a trait which is slightly different from the trait from the economic spectrum. This is a leaf dry matter content, which is the dry mass, water saturated fresh mass, and which indicates the tissue density, the density of tissues, and which is one of the components of leaf mass covering. When you have a low leaf dry matter content, you have soft tissues, a high proportion of basophil at epidermis, and a low amount of fibers. And when you have dense tissues, you have a high proportion of sclerenchyma, vascular bundle, and a high concentration of fibers. So with this, combining this data, we calculated 2 community metrics: community-weighted mean, which is the central tendency of the community, and community-weighted variance, which gives an idea of the functional diversity of the. And then we measured dry matter digestibility on the green bulk standing biomass. So we took 1 square meter of the different communities and measured the digestibility of this bulk, the biomass. So if we look at the response part of the framework, when you change the land use, when you change the management in the stuff, what happens in terms of leaf dry matter content? So that's a community-weighted mean, which is the sum for all the species which are measured of the relative abundance of the species times the value of the traits for this particular species. So when you calculate community-weighted mean of leaf dry matter content in ungrazed, when grazed and ungrazed part, the leaf dry matter content is higher as compared to fertilized and highly grazed plots. So it means that you decrease the leaf dry matter content as you increase the management intensity. If you measure the dry matter digestibility, not of the whole community this time, but for each species, which are measured the same species as here, you still have a decrease in digestibility. So the species that are in the fertilized plots are more digestible than the ones which are in the ungrazed and grazed fertilized plots. So from this, I'm going to test, and this is what happens to the dry matter digestibility of the bulk community biomass. So this is the digestibility of fertilized plots is higher than the other. So now I'm going to test the dominance, the selection hypothesis. What I want to see is predict is the digestibility of the bulk community biomass, 1 square meter, from the species represented. So the first thing that we did is that we compared the calculated community-weighted mean of the species leaf digestibility of each species, and compare this to the community digestibility as a whole, you can see that there is a relatively tight correlation. There is a change of the, it's not exactly the one line, but still the relationship is pretty tight. Now, if digestibility is not a very easy trait to measure, you have to have a near price spectrometer and stuff like that. You have to change, if we had a trait easier to measure, that would be good. And so we tested the idea of the relationship between live dry matter content and digestibility. So this is at the species level. This is the relationship between leaf dry matter content at the species level and the dry matter digestibility of this species, which is very tight. And then if you scale from leaf, the community weighted mean of leaf dry matter species to community digestibility, then you have a very strong relationship. This means that in a community with a low light dry matter content, fertilized plots have a much higher digestibility than fertilized. If I do the same test for the complementarity hypothesis, then I calculate another metrics, which is the community weighted variance, which is the sum for all species measured of the difference between the mean trait of the trait of the species, minus the community weighted mean of trait, the average weighted by the abundance of the species. Then this is what you have. What we have is that the variance, the community weighted variance, so the differences in terms of traits between the species decreases as you fertilize. When you fertilize, the community tends to converge to a common value of leaf dry matter content as compared to the other ones. So if you plot the relationship between community weighted variance to leaf dry matter content, then you have a negative relationship. It means that the community weighted, the digestibility is higher in less diverse, functionally diverse communities. So we can ask whether this is simply a fact of this particular thing. No, sorry, two things. So maybe I'm going to how I'm showing the new type. Okay. So we actually generalized the test of the dominance hypothesis across sites, so the 9 sites with permanent grasslands, which are all across France, in which we had 120 plots with different disturbance and fertilization regimes. And what we measured, what we showed is that for each of the sites, you have so the 9 sites are here, each of the sites, you have the same relationship as we had for Lafarge. Lafarge is the red one here. And we have the same type of relationship for all the sites. But you have a shift in the intercept of this relationship. And we showed that it's actually related to the climate of the sites. The drier sites have the drier sites, so the PT is the potential evapotranspiration of the site. The drier sites actually have, for the same community weighted mean leaf dry matter content, have a lower digestibility. So we established this equation from these graphs, and we used it as a functional biogeography exercise during one of the first project led by César, which is called DeepGrass, in which we compiled data. Cyril was the postdoc of this project. So we compiled data from 50,000 vegetation rows across France, took leaf dry matter content from the dry database and the potential evapotranspiration from Météo France, and then we mapped we have a map of the digestibility of French permanent grasslands, showing, for example, that the digestibility of communities which are in the north of France, in Normandy, are much higher, for example, than in the Pyrenees or in the Alps. So we have a map now of this kind of, we can do that map of this kind of properties with the traits. So just to sum up this, so what is the conclusion of that? So for a book we have published a couple of years ago, I made a literature survey, which had actually tested the two things, whether ecosystem properties were linked to dominance or complementarity. At the time, there was 25 studies which had simultaneously tested dominance and complementarity effects. And so far, number of trait combinations, I'm not going into details. But anyway, what we see is that in almost 90% of the cases, the community average had a significant effect on ecosystem properties. And in 60% of the cases, functional diversity had a significant effect, sensu stricto, in functional diversity equal either negative or positive. But to test the complementarity hypothesis, this functional diversity effect has to be positive. So you can split the two. So here you can see that in 40% of the cases, the effect of functional diversity is positive. So it means that 40% of the cases, the complementarity hypothesis is verified. And the functional diversity effect is negative in 20% of the cases, as I showed you for the Lafarge. So it means that in 80% of the cases, the dominance effect is verified, and in 40%, the complementarity effect is in 20% of the cases. So the conclusion is that the functional structure of connectivity influences ecosystem properties, that recursive traits scale to the ecosystem level, and that there is currently more support for dominance than for complementarity effects. It doesn't say that complementarity effects are not important, but from the literature, the dominance effect is. So now I'm going to switch to the last part, which is the part easier, which is making sense of the data. So here I'm going to take the example of traits, but I think you can think of your own data. What I'm going to say is very generic. We have applied it to the trait data, but it's extremely general. So during my talk, I actually showed you results from large databases, which are TRI and the DeepRAS databases, which are actually a combination of small data sets. So these big data sets come from small data sets which have been aggregated. And there are high values. I think now there's currently more than 15,000 requests to try database to get traits. So it's highly successful. Those databases are highly successful. But we should not forget that these big databases come actually from small data sets, which have their intrinsic value by themselves, because if people have done experiments or made observation and they think that their value, the data set is valuable by itself, so their intrinsic value, and some of them have put these data in the big data sets. So the idea is that, is this easy? If you have the Excel file, you're happy with your Excel file, you send your Excel file, that's fine. You think that's the case? Well, good. And one of the main issues is that the degradation, the information which is contained in the dataset degrades all the time. The problem is that, for example, you have think of your own data. When I say that, think of your own data. Is it information content of your data? When you do the experiment and when you publish your data, you know everything about your dataset. You know all the details. You know how you collected the data. You know how you have formatted that in Excel file. You know the units. You know everything. Then think of yourself 5 years later. You've published, your dataset is there on your computer, and you want to go back 5 years later. If you have a very simple Excel file without any information, then you're going to be in trouble. You may remember a few things, but probably not everything. When you retire, which is my case, then after retirement, it's even worse. I mean, when people are retired, it's very difficult to contact them. If you have a simple Excel file, you won't locate it. And if you die, it's even worse. So you may contact some retired people, but probably don't die. And there is a solution to that, which is actually the data to implement the data life cycle. So what you do usually is that during a very classical experiment is that you plan the experiment, you collect the data, you check the quality, and you analyze. And then it's done. Actually, there's two important parts in this data life cycle, is that you have to, if you want to reuse your data, and even if you go back yourself 10 or 15 years later, if you want to reanalyze your data, put them in the broader context, then you would have to close the life cycle by describing and publishing and preserving the data properly. That's the idea. So again, we are in the world of open data, and this goes with open data. But open data is not just giving away your Excel file. It's really to apply a number of principles, which are called FAIR principles. And this This paper was published in 2016, so and it had more than 2,000 citation currently. And the idea is that you have 4 principles to follow. Your data should be findable, accessible, interoperable, and reusable. I'm going to go through that later on. And it imposes, at least for ecology, a complete change in perspective on your data. You should switch from when you think you are the owner of the data. It's true, obviously. But you have the responsibility to give away your data. We are public servants. We are paid by the government. Then your data should be open to the others. And it's a very strong switch from you think what you own the data from what we call data stewardship. So you should switch from this attitude to a wider and broader attitude. And these are very general principles. But when you try to apply them, then comes the issue. So we've tried to do that. And the word try is also try. And we've tried to do that for a database that we have for trade data collected by the Ecobar research group at CEF over the 1997 to 25 years, basically. And it was just published in Ecology as a data paper. And we tried to apply these different principles on this database, which is consisted of 180 traits, which were collected on 6 sites on 240 species. And we have a little less than 200,000 records of the database. And this gives you the range of traits that we have in the database. It's much wider than the 3 traits we've been talking about, 6 traits that we've been talking about. Anyway, how did we proceed? So I'm not going to go through all this, but we applied this fair approach to this. So the idea was that the data should be reusable, interoperable, findable, and accessible. Not going to develop that too much. But I think there was one step which is missing in the fair approach, which we call the data should be meaningful. And if you do all this without meaningful data, then it's just useless. Don't do it. And the idea is that we propose to introduce this step and going from fair to, I'm not very good in acronyms, but to mega. But it works. It just needs M, A, G. Just miss the A. But I think we can try. Anyway, I'm going to show you what we did for our full database. So if I give you a file with an LDMC value equals 200, and let's just say that. Don't think you can do much with the data. First, you should have the unit first. But anyway, we know already that this trait, this particular trait, varies with a number of things. It varies with the natural history of the crops. This is the analysis of the fair trade database. For example, if I compare the leaf dry matter content of woody and non-woody and herbaceous perennial, then there is already a signal in the value of the trait. So it's a good point to put some stuff on life history of your database. It's quite interesting to have taxonomy as well. Here you have the leaf dry matter content of Asteraceae, composite, and poaceae, grasses. And you already know that there are huge differences in leaf dry matter content between Asteraceae and poaceae. So you should at least put something on the taxonomy in your database. Then you know, and this is one of the things we are studying, is that there is a relation, an effect of the abiotic environment on the traits. This is the differences in leaf dry matter content between high and low nitrogen. Not big, but the difference is significant. You can trust me, yes or no, but anyway. So these are the minimum. We list quite a number in the paper in preparation with a video. We list a number of things which are interesting to have in the database to interpret the trait value. And the key point is the methodology. So you have to have something about the sampling strategy and the measurement methods. But what I'm going to focus on now is actually the fact that you can model traits from a conceptual point of view, and we use that from the fields of genetics, where the phenotype traits are modeled in the entity quality models. And the idea of the trait is the quality of an entity. The quality would be what you measure: the area, the mass, the length, anything. The entity would be the part of the plant on which you measure the quality. The leaf, the height, the the whole plant, the roots, etc. So you have two parts of the traits, which is modeled with this. And why is this important? Imagine that you take the example of roots, because Catherine was worked a lot with roots, and she has worked a lot on what are the different functions of roots. And some function of roots, some roots actually absorb nutrients, and they have a number of traits. And some of the other roots actually transport the nutrients and water through the plants. And actually, the traits of these two type of roots are very different. If you look at the specific length, which is the length of the root per unit mass, actually they should be considered as different traits. And this is what we did in the database, just to show you the specific root lengths of absorptive, so these ones, and transport roots. There is a tenfold difference between the two. So if you just say I'm measuring the specific root length of the root, then you're stuck. You don't know exactly what you've been doing. So the idea is that to be very careful in what you are doing with the entity that you're measuring the things. So I'm going to go quickly through the other steps. So and the other important thing is that you have to use standards and relevant repositories. So you have to use standards for terminology, how to define your traits, how to define them very properly so that people can understand what you have been doing in terms of semantics. You have to use protocols for measurements when they are available. And if they are not available, the idea is that you actually describe the protocol in the database. So you have specific fields to describe that. And then you have to describe properly a whole bunch of things which are called data and metadata. Metadata is everything that you think is important to understand the context of the data. And another thing is that depending on the repository that you are going to choose, you can choose a standard for the data itself. And for Fairtrade, we use the Darwin Core standard because actually we put the data in the GBIF facility. And you can only upload data in GBIF only if it's formatted through Darwin Core format. We use an extension of the Darwin Core, which is currently in process, which is the Billion Core. And an interesting thing is that when we were doing that, we realized that Darwin Core was not properly describing all what we wanted to do. And we had exchanges with the GBIF, and they are trying to change the standard Darwin Core standard so that they can incorporate more and more data, different type of data. Okay, and the idea. So remember the first curve I showed, which is this, and Nicolas Mouquet, who is a very optimistic guy, said that if we do that, the quality of the data can increase after a colleague's evaluation because you have produced a database, for example, or a data paper, for example. And even at the data, another very important thing is that when you merge your data with other data, you can see your data from another perspective. And you can, even something that you could think, for example, was an outlier in your data set, actually is not an outlier, it's just an outlier in your data set, but not general. So this is very important. And maybe future technology might have some other problems. So to conclude quickly, so we switch from a very broad definition of the biodiversity in 1998, when I was 5, to something which actually we know now how to describe much better what is behind the biodiversity from the cell, genomes, to species, traits, population, community, ecosystem, even to the planet. Biomes are considered biodiversity. There is now a relatively clear framework of how to understand the pressure on biodiversity, different components of biodiversity, and the consequences on nature and ecosystem services. This is the work by Bess in particular. Bess actually did a very strong report on that. And then corresponding to this, we actually a whole bunch of databases. It's just amazing when you look at the databases which are available now. It's just huge from CBI data, from genomes to dry contracts to GBIF, which is basically taxonomy. And you have databases for biodiversity stuff, but also for the pressures that you can see here, and for the impacts of the different pressure on biodiversity. And if each of these databases works separately, I don't think we are going to be able to answer big questions that are asked to our discipline. So I would like to end this talk by to thank my numerous collectors over the years and networks, and especially the EcoPar group, which I created in 1988. And these are the different avatars of the group. I really like this one in the middle. I think it's really the 3F group, research group. It's very friendly, very fruitful, and very stimulating. And before I go to another path, which is probably one of the ones that you showed in the slide, I'll be happy to answer the questions.
Thank you, Eric. I know we are a bit off over the time, but it's fine because that's his responsibility.
Okay.
But we'll take questions. There's an ask to speak loud because the microphone is just in front, okay? And we'll also take questions online. Yeah, so please go for it.
Yes. Maybe the first detailed question, of course. Thanks for this very nice overview from really a specialist. So why, detailed question, why is leaf area linked to fecundity? Is that a direct, is there a direct causal effect, or is it because there are underlying factors?
No, I think leaf area is really the amount of leaf that you have on the plant. So it's basically the size of the plant. It's not the area of the single leaf. It's the area of the whole canopy. So it's just sun. But I couldn't find the data just showing this for biomass or any other size. So it's resized. Yeah. Staying there because we can also see you. Yeah, it's better. We're planning for it.
So I would say that this whole enterprise of collecting and measuring traits has really informed us about syndromes of traits and how traits could vary. But I think the aim was really to get at functional ecosystems and to get this atmosphere. And so what would you say? You identify things that really we can predict with these traits at the scale quite efficiently, and other things where the enterprise failed and we can't predict as much as we were hoping by collecting this huge amount that are on those traits that are easy to collect. Can you say what you think are the success and failure of this?
Okay, the main issue I see, there are several things. That's a very big question, actually. I was expecting that from you, definitely. Yeah, there's a whole range of things. The first thing that maybe is the fact that to sort out the biotic and abiotic effect on ecosystem functioning, because all the experiments that have been conducted experimentally on small plots or what I've been doing are really local experiments. If you scale that up, you may end up with having the abiotic effect, for example, temperature or water availability, which are more important than the biotic effect by themselves. So this we don't know exactly. So this is one of the things we should really sort out, whether biodiversity by itself is important or whether large, over large gradients is other component important. So that's the first thing. The second thing is that there is a mismatch very often between the traits we measure and the ecosystem process we are looking at. I think we need a better mechanistic basis of why we connect a certain type of traits to the ecosystem process at hand. For example, I showed data for leaf dry matter content because we knew that this particular trait is linked to fiber content, and we think that it's important for digestibility. But when a lot of literature take just the try database with any traits and try to scale from very simply from any trait that there is in try to ecosystem processes, just getting to fishing what is important or not. And I don't think that's a very good approach. So linking the process by the ecosystem process that you are looking at to the actual traits that are important. If you look at water uptake, for example, you're probably looking at more important to have rooting depth than specific leaf area. So this is another point that should be looked at. And the other thing is that
actually to disentangle the community with the average effect to diversity effect is very tricky because the moment of the distribution are related to one another. So you actually cannot disentangle properly the two things. So you have to design experiments that manipulates one or the other separately. So that would be maybe the main things that I would put forward for the scaling. But I don't agree with your first sentence, because actually we've been looking also at the response part. And for the response part, the trait might be different. And again, we have to look at the actual mechanism which is behind the response of the plant to the environment, and look at the proper traits as well. So if you look at the gradient of nutrients, the traits that are going to respond are not the same as if you are going to look at a shade gradient. And you have to be very selective on what you are doing to try to approach the mechanism which are behind. Is that okay? No?
Just une autre question, Vincent. Alors, oui, peut-être avant toi, enfin, comme vous voulez.
Thanks for the very good talk. I'm really struck by this figure that I've seen before, where you use the construct data, where you see that the spread of the traits just for a redox is basically broader than anything intraspecific that you have based on the mean, at least for the core distribution. So this really gets me thinking. So you say it's a very special species.
It's not as
Oh, I said Arabidopsis is amazing, and it's recorded.
It's been studied a lot, basically, and so lots of these variations, geographic variation is ecotypes, I guess. So I don't know what my specific question is, but basically, lots of the within species variation could be strong, spanning more than the within species. So if it's if it's local, then it means these species interact with all these variants of the other species, so it's going to be important. If it's geographical, then you need to account for it, but it also says that it's also a potential for rapid evolutionary responses within the species. So I don't know how you can include that.
maybe I can, François, you can answer this question, because I think you're the one to answer.
So this data is one of the models. So the code the GLS are going to inform the world. So if we have the genetic of that's a big deal. I'm not sure it's really specific for biodiversity. I'm sure species can have the same range of information, but I totally agree that it's a very nice reservoir of biodiversity for evolution. So yes.
Just if you had this for each species, then the between species thing would change a lot. Even the sports.
Yes, yeah. One key point what you say is that using an average is very dangerous for the species. So depending on how much genotype you use for characterizing the species, you can have very different location of the average species in using this graph. So that's why, yeah, it's important to take care of just.
Yeah, maybe to complement this question. So this is the relationship for not for the whole space, but for the economic spectrum. And the very important thing is that everything is log scale with this analysis. So it's not completely innocent. And if you look at so you can look at the scale of net photosynthesis for olive trees and for Arabidopsis. The range is very different actually. And even the range for each trait, each individual trait is very different. And actually, it's the spread of the data is amplified because Arabidopsis has low values of the traits as compared to, for example, the olive trees, which high values of the traits. So actually, you distort the amount variation of Arabidopsis as compared to the other species. So that's log scale is a bit tricky in this.
But perhaps meaningful to make those. So it goes back to meaningfulness.
Yes.
Anyway.
Yeah, log scaling is really, yeah. Anyway.
Joss, après Etienne.
Yeah, so maybe a less trivial question. So in describing diversity, we're often confronted with species that are very, very dissimilar from other ones. So that you can't measure traits on I'm interested in plants, so one species I'm interested in is Rafflesia. It doesn't have leaves, it doesn't have stems. So what do you measure? So is there a way to incorporate these species in this biodiversity framework and ecosystem functioning, or are they just too rare to care about?
No. Well, I mean, that goes back to functional variety, which is another issue. But yes, in terms of traits that you can measure, I mean, we measure leaf mass area on nitrogen because we know that's light interception. So what would be the light interception organ in your particular species? That's what you have to measure.
But it doesn't.
Wait, it's an autotrophic species. Is it?
Yeah, it's a parasitic species.
It's a parasitic species. Okay, so there's no chlorophyll, no everything. So I would turn to the actual organ that capture carbon. The equivalent would be, what is the organ that capture the carbon for you? You need carbon, you need nitrogen, you need stuff. What is the organ which captures the carbon? There is someone. There is one. There is one. Your skeleton is carbon. So there is one obligate trait that would tell you something about how the plants absorb carbon. So that's really the way of thinking, is that what is the traits that what is the process that you're looking at? And about this, when you know the process, look at the traits which is relevant for that. So it might be the case that if you do that, you cannot compare that at the broad scale that as we did, but it might not be an issue. It's just the way your plant works, and it's not there. Etienne?
Thank you, Eric, for a very inspiring talk and your contributions over your whole career. It's been very inspiring to many people in this room. So I was curious, as you're nearing the end of your career, obviously you've shown about the promise and the success of the functional ecology approach, but there are also limitations. As every field evolves and there's new directions, what do you feel after 35 years right now?
You have 2 minutes.
What interesting new directions for the future of functional ecology?
I asked you this question a couple of months ago, and you said, pfft. And we asked Peter Rice yesterday the same question. He said, but anyway, I'm going to try this. Because actually I've prepared a slide.
This was not staged.
And you are going to be afraid. Okay. I think this is the main limitations of what we've been doing. So maybe I don't go through it a bit, but I think one of the key things that we have is we have worked a lot on traits. We know how to describe them, we know how to measure them, a whole bunch of stuff on that, but we are very bad at quantifying the environment. Where the environment of an organism is, I mean, it's always almost a black box. You don't know what nutrient availability is, how you can find that, what water availability is, temporally, especially, what the temperature that is sensed by the organism is. We are very bad at it. So if we want to do something, I think we really have to a better description of the environment that we're looking at. And we have also more or less the same idea, that we have to quantify the relevant environmental variable you're looking at. You know this literature, when you plot the relationship between any kind of macro scale idea of temperature or precipitation or whatever with the traits, actually the x-axis is not the right one. So which one do we do? So that's very important. For the response to the environment, yeah, we have a number of questions to solve. So we are a little bit far away now from the ecophysiological basis of the response. I think that's probably one of the things that should be thought of much more precisely for the plants. I mean, LMA is not going to do the whole trick. We need something else. For community structure, I don't know if you know this paper by John Lawton in 1999. It said community ecology is a mess. And I think I'm going to, maybe can I say, I'm retiring, I don't care. I do think community ecology is now a mess, but for other reasons. I think the idea of how species coexist with the trait-based analysis, which is very promise of the package paper in 6. I think this has failed. I think there's too much statistics. You're much far away from the biology, and I think it's not going anywhere. That's my feeling. I know people are not thinking the same in the room, but I don't trust these papers and these analyses, basically. And Francesco De Velo has a very nice example of that. Is that when you try to understand whether the species converge or diverge in communities, you have a succession of filters and models that you have to use, null models and filter model. And depending on the pool of species that you take regionally that you don't know, depending on the pool of species, the outcome of the model is different. So I think it's really becoming a mess as well, but for other reasons, as John mentioned. And then for effect traits, I answered a bit for Montpellier, and this is the list of things that needs to be done for effect traits.
Roger, il y a des questions en ligne aussi ou? Ok, bah Philippe, alors vas-y.
Thank you, Eric. More matter. I mean, yes, there was this paper by Rottens and community ecologies and best. I mean, we have a lot of theory, we have good theory, it's different of species, and I don't think we should converge to one single theory. In fact, I don't believe in this. And that's probably an error to believe that, for example, physics, we are always compared to physics. True, we don't do that. But I was perhaps going off what this was saying. I guess that we are in entire room, and one of the main ideas of Montpellier was the other virus sites of the study. Wasn't talking about biodiversity at the time. So paleontology, which gives you some about but also what's going on now, and to develop a true eco-evolution. And I guess that we have made huge progress in this direction. I'm old enough to remember this difference going on between 70s and 40 years ago almost. And to see what, for example, François is doing, using explorations, and what the other people in this are trying to do, going from observations to more community perspective. I think we are going in the right direction. We are trying to build a continuum, true continuum of what. And I think that these are the right directions in which, as a community here in Montpellier, we should proceed. And I think also that that's a unique place to do it, and we are contributing to that. And
The still community ecology is the best.
Maybe,
Thank you.
thank you. Maybe last question from Roger.
Yeah, just was curious. Thank you very much for this presentation. Was just curious if this big database also included the submerged aquatic plants. Because I would expect that they behave, of course, very different with respect to CO2 uptake.
In the trial database?
Yeah.
No, there is no macrophage.
Okay.
No. But actually,
this type of approach is happening.
yeah, it does. Actually, Jules Segrestin, who was a PhD student with me a couple of years ago, has been working in Lyon with Sarah Pujalon, and he's been doing gas exchange in submerged plants, and he's been looking at whether it would fit, yes or no, on the global spectrum. And apparently, it's not too far away from the leaf economic spectrum. So even the traits of the submerged plants seems to be something pretty close to what they have in the aerial system. I don't remember exactly how they are in the spectrum, but still it's consistent with what we have for terrestrial plants. Yeah, I should check with Jules, but I think that's more or less what we have.
Yeah, thank you.